Recall when we are inside the small intestine and complete the digestion of triglyceride molecules, we see that mostly triglycerides are broken down into monoglycerides and fatty acids. Monoglyceride is one glycerol attached to one fatty acid, and then the fatty acid molecules that we get are classified in two groups. We can say we have lightweight fatty acids. These are fatty acid molecules that they have about less than 10 to 12 carbon atoms, and the absorption of these small lightweight fatty acids would be different from monoglycerides and long heavy chains of fatty acids. So here in this image, we're looking at one of the absorptive cells that line the small intestine, and we want to know exactly how this small, short, lightweight fatty acid passes through the apical surface of this cell, and then from the cell passes through the basal surface to get into the fenestrated blood capillary that we have in the connective tissue. So the point here that we're making is that if we are absorbing short chains of fatty acid that they are small and lightweight, they are absorbed into the blood capillary directly. These short fatty acids are water soluble, and very easily through simple diffusion they pass through the apical surface, and then again through simple diffusion pass through the basal surface and get absorbed into the bloodstream. So in general, we do not need to use any ATP hydrolysis, any energy to absorb these short lightweight fatty acid molecules into the blood that we have here running through the fenestrated blood capillary. However, when we focus on the absorption of heavy, long fatty acid chains or monoglyceride, we see that, first of all, these molecules are hydrophobic, they're not water soluble, and they have really difficulty to stay in the watery kind that we have inside the small intestine. Keep in mind, we are in the lumen of the small intestine. So we get help from those components of bile that are amphipathic. Those are bile salts. Recall that in each bile salt, we have a region that is hydrophilic, water-loving, and we have another region that is hydrophobic. This part, the hydrophobic end of the bile salts, can easily interact with these heavy long chains of fatty acids and monoglyceride, and exactly what bile salts do in the lumen of small intestine, through their hydrophobic end, they interact with these lipid molecules, and basically the bile salts form a coat around the monoglycerides and long chains of fatty acids. Usually when we look at this structure that they form, about 20 to 50 bile salts wrap around several of these monoglycerides and long heavy chains of fatty acids and form a structure that is called micelle. So micelle forms in the lumen of the small intestine, and the coat of micelle is made of bile salts. Inside the micelle we can find monoglyceride and heavy chains of fatty acids. What micelle does, it actually helps these hydrophobic molecules to be transported toward the microvilli of the absorptive cells. So think about formation of micelle as a strategy that bile salts use to transport these hydrophobic, large, heavy molecules toward the microvilli of the absorptive cells to allow them to be absorbed into the absorptive cell. And we can see that when micelle get close to the microvilli of the absorptive cells, the inside content, which would be monoglyceride and long heavy chains of fatty acid, by simple diffusion get inside the cell. What about the bile salts? The bile salts go back into the lumen to wrap around more of these molecules, form basically micelle, and again transport them toward the absorptive cells. So that would be the role of bile salts, to keep forming micelle in the lumen of small intestine and bring these hydrophobic lipid molecules to be absorbed into the absorptive cells. Now, when we focus on what happens to these monoglycerides and long heavy chains of fatty acids, it is interesting that inside the absorptive cell, we see that we have enzymes that allow fatty acids to bind to monoglyceride and form the large molecule of triglyceride. So triglyceride forms inside the absorptive cell, and then we can see that triglyceride molecules basically aggregate, they get together. That's basically the habit of hydrophobic molecules such as lipids, that they tend to aggregate and form larger globules. And also we see some other lipids such as phospholipid and cholesterol are added to the group, and then the absorptive cell basically prepares these molecules to get inside a watery place by producing some proteins and wrapping a coat of protein around the triglycerides, cholesterol, and phospholipid. We call this large compound that forms inside the absorptive cell chylomicron. And then the absorptive cell uses its organelles such as endoplasmic reticulum to pack the chylomicrons inside some vesicles, and through exocytosis pass them through the basal surface. Recall, exocytosis is a vesicular transport, and it's an active type of transport. It simply means that the absorptive cell has to use the energy released from ATP hydrolysis to do the exocytosis of chylomicron. Chylomicron is very large. It cannot be absorbed into these fenestrated blood capillaries that we have in the connective tissue. So we have to use a more permeable capillary, which is the lymphatic capillary. And recall that we named the lymphatic capillaries that we have in submucosa of the villi of small intestine lacteal. So we see that the chylomicron gets inside the lymph that we have in lacteal, and we know that lymphatic capillaries join together, form lymphatic vessels, and eventually lymph from small intestine gets inside intestinal trunk. And we know when we follow the intestinal trunk releases lymph that contains chylomicron into thoracic duct, and we know that thoracic duct drains lymph back into the venous system near the junction of left subclavian vein and left internal jugular vein. So the difference here is that if we're absorbing a short, small, lightweight fatty acid, the absorption is entirely passive and would be directly into blood. But when it comes to heavy chains of fatty acids or monoglyceride, as you see, we have to form micelle by getting help from bile salts in the lumen of small intestine, and then inside the absorptive cell we form the triglyceride that aggregate with phospholipid, cholesterol, and proteins that are built by the epithelial cell to form chylomicron. And then through an active process, exocytosis, chylomicron has to go inside lymph, and then lymph is drained into blood. So the steps that we require to absorb these large molecules of lipid would be more number of steps and would be more complex than absorbing short, lightweight fatty acid molecules.